Hello, welcome back. This is about the bacterial heterotrophic nutrition. The source of carbon from the external organic compounds. It may be two types, photoheterotrophs as well as chemoheterotrophs. Photoheterotrophs means carbon they are getting from the external organic sources and energy from the sunlight. So we call it as a photo. Hetero means depending on the others, so they can get the anyhow the organic sources. They are non also pigmented because they observe the light energy. They need the compulsory the pigments, and they are a purple green non-sulfur bacteria, rhodospirillum and rhodomicrobium. Chemoheterotrophs, chemicals they are preparing by depending on the organic compounds. They are getting carbon and energy from the organic compounds. They are non-pigmented. Chemosynthesis will be done. Saprophytic, dead organic matter they are depending. Example bacillus. Parasites they live on the host. Example salmonella typhi which is causing the typhoid. Symbiotic, a mutual association with the higher plants. Example rhizobium bacteria with leguminous plants, which is helping for fixation of the nitrogen. So we call chemoheterotrophs and photoheterotrophs. Carbon and energy from the external and as well as sunlight energy. And here carbon and energy from the organic compounds they are getting. So that's all about the heterotrophic nutrition. Let's see about the reproduction in the bacteria. Generally asexual reproduction and sexual reproduction. Sexual reproduction is like a genetic recombination, and asexual reproduction without fusion of male and female gamete, production of the new organism is called it as an asexual reproduction. Here we see binary fission occurs by means of the direct cell division. Binary fission during the favorable condition. This process we can call it as a amitosis because there is no spindle fiber formation, there is no separation of the chromosomes. Circular DNA replicates, plasma membrane invaginates, that means enters inside, which grows centripetally from outer side to inner side, divide protoplasm into two parts, becomes the double. So this type of a nature we call as a binary fission. Though two molecules there, two bacteria they are getting formed here, and this process approximately for DNA replication it takes 18 minutes, and it depends on the bacteria to bacteria, variations do occurs. So endospore formation it takes place during the unfavorable conditions. It is highly resistant to the temperature. Spore is formed inside to tide over the unfavorable conditions. Perennation, that is ability to survive more, also will happen. Exosporium spore coat cortex and core wall. Calcium dipicolinic acid will be produced. They are thick and impermeable spore coat, low water content and low